So let's see. Okay, I think we can begin. So today I'm introducing Robyn Wright, who is right now in Nova Scotia, and she's doing a postdoc at Dalhousie University in the lab of Morgan Langell. And she is interested in the roles that microbial communities play in human and ecosystem health, such as, for example, studying environmental reservoirs of pathogens, like, for example, with the biofilms of marine plastics, which she's going to talk about today, I think, or also isolating and characterizing bacterial strains that are potentially able to remediate human or ecosystem health threats. So Robyn, I think I'm going to give you the microphone. You can share your screen.
Thanks. Okay, so can you see that?
Yeah, it looks good.
Great. So hi everyone, and thank you for the introduction, and thanks for inviting me to talk today, and to everyone that's listening here. I'm going to be telling you a bit about the Plastisphere and what we know about its role in marine plastic degradation. But first of all, I'll just give you a bit more of an overview of who I am and the projects that I've done to lead to this. I'm currently a postdoc in the Langell lab at Dalhousie University in Nova Scotia, working on the human microbiome. And prior to that, I was funded by the Association of Commonwealth Universities Blue Charter Initiative for a 6-month project that I carried out also at Dalhousie University with Tony Walker and Morgan Langell. So I'm going to be talking about the meta-analysis project that I carried out during those 6 months, but I'm also going to be talking about some of my PhD work that I finished at the end of 2019 at the University of Warwick in the UK with Joseph Christie-Laser and Matt Gibson. And that was funded by the Midlands Integrative Biosciences Training Partnership. So I'm going to run you through some of my work that's been on marine plastic pollution and the potential for the degradation of these plastics. And I'm going to tell this story in what I think is the most logical order, although this is not the chronological order of the work. So if you've seen the news with any regularity over the last few years, then you'll be well aware that the oceans are now full of plastics. And once in the oceans, these plastics experience a range of different processes. For example, they can be transported long distances by wind and currents. They can be fragmented by wave action. They undergo weathering through processes such as UV photodegradation. They can be ingested by a whole host of organisms, and also they can be colonized and potentially biodegraded by microbes. The microbes that colonize plastics in the ocean are an often distinct group of microbes that are known as the plastisphere, a microbiome for plastics, if you will. No one really knows definitively what the metabolic capacity of the microbes that make up the plastisphere is, but it's been suggested that they're just surfing on the plastics or along for the free ride, that they're pathogenic and maybe traveling larger distances on the plastics than they would otherwise, and also that they could be degrading the plastics. So the plastisphere and marine plastic pollution more generally has been really heavily reviewed within the last year or two, and this is not even a comprehensive list of these reviews. And if I focus on one of the reviews that I helped to carry out, then this tells us that plastisphere studies have used a range of experimental and methodological conditions to characterize the plastisphere. So here we're just looking at the methods used by each study and not the results of the studies. And we can see that we have a range of different incubation setups, ranging from collection from the environment after an unknown residence time to a time series of incubations. We have different next generation sequencing or microscopy techniques used for characterization. Studies have controls ranging from planktonic to control biofilms. Some studies have aimed to look at pathogens and antimicrobial resistance genes, and others have used various measures to try to quantify plastic degradation. And if we take these results together, then these studies have concluded that members of the plastisphere are different from communities that colonize other surfaces, not different from communities that colonize other surfaces, only different from communities on other surfaces under specific environmental conditions or only at specific time points, that they're more diverse than other microbial communities, they're less diverse than other communities, they're potentially degrading the plastics, potentially degrading the additives of plastics, and that they're potentially pathogenic and or carrying antimicrobial resistance genes. But how do we know which of these are true or in which circumstances? So put simply, what we really want to know is what happens to plastics in the oceans. Are they being degraded? And if so, who is degrading them? So I'm going to divide my talk into 3 sections, and hopefully I can shed some more light on this. I'm going to be talking about what we already know about the plastisphere, or what knowledge we can gain from reanalyzing all of the plastisphere studies together, whether plastics are being degraded in the oceans, or perhaps the more pressing question of whether they even can be degraded in the oceans. And then finally, I'll be looking at whether the additives and contaminants of plastics can also be degraded. So at the beginning of this year, there were 27 studies that used next generation sequencing methods to study the marine plastisphere, and 50 studies if we also include freshwater, aquatic and terrestrial environments. And as we can see on this map, all of the studies were carried out in the Northern Hemisphere, the vast majority are carried out in temperate environments, and a large proportion of these studies were conducted in and around Europe. So while these studies are all theoretically comparable, they use different methods for analysis of their next generation sequencing data, such as ASV versus OTU and rarefied data versus relative abundance. So if I have anyone listening that's not familiar with these techniques, then the point I'm making is that they use different computational methods for analysis, which makes them not directly comparable in their current form. So we collected all of the raw sequencing and metadata that we could, meaning that we could include 23 of them for the marine environment and 35 in total, that either already had accessible data or made it available upon request. And while a couple of these studies used shotgun metagenomics and several of the studies looked at the eukaryotic or fungal communities, we didn't really have enough there for a meta-analysis. So I'm focusing here on the prokaryotic or 16S communities that were determined through Illumina amplicon sequencing. And the first step after gathering all of the data was to process it. And for this, I used a fairly standard QIIME2 workflow. I did test different methods for normalizing these data, but if you're interested in that, then I'll have to point you towards the paper. The thing that was different that I did was I used an agglomeration step to try and deal with the many different primers used by these studies. So basically I inserted all of the amplicon sequence variants into one of the silver trees, and I then agglomerated all of the sequences that were less than a certain phylogenetic distance away from one another. And because this is phylogenetic distance and not, for example, percent sequence similarity, it's a bit difficult to say exactly what rank this corresponds to, but it's generally somewhere between species and ASV. So I've continued calling these taxa amplicon sequence variants or ASVs, but when I say that, I'm talking about these agglomerated ASVs. We then categorized each sample to a group within each of 20 metadata categories, which included things like temperature, salinity, light availability, geographic location, primer parent DNA extraction method used, the study that the sample came from, and the substrate type to varying levels of specificity. And what we wanted to know was which of these can most accurately predict microbial community composition, and then which taxa are the most informative for this. So to do this, we constructed random forest models on each of our metadata categories. And we also wanted to know whether certain taxa are consistently associated with different plastic types. And I will go into the types of plastics this includes later on. And also whether there are taxa that are more abundant at early than late incubation times and vice versa. So along with the random forest models, we also carried out differential abundance testing to find this out. So to answer that first kind of overarching question of which of these categories can most accurately predict community composition, these are the results for each of our random forest models, which contain all samples, even those from different environments, and then constructed for different metadata categories and in each phylogenetic level from phylum to ASV. These are based on an 80-20 split of our data, where we use 80% of the samples to train the model and then 20% for testing. And the numbers in the boxes are the percentage of the time that our model can accurately classify the sample to the right category within that metadata category. So for example, within the laboratory or the field category, we're looking at how often our model can accurately classify a sample to whether it was from an experiment conducted in the field or in the lab. And here the lighter yellow colors mean that the classification was good. So these categories are sorted from most to least accurate. Top to bottom, and the best performing random forest model was accurate 94% of the time, and it was trained on the lighting regime. So whether the samples were incubated in ambient or modified lighting conditions, such as in total darkness, and this was most accurate at the class level. Other categories that performed well were things like the environment that the sample came from, the primer pair used for sequencing, and whether the sample was incubated for a known length of time or collected from the environment after an unknown amount of time. And then we see that while our plastic type categories aren't the worst, they aren't performing very well either. they're less than 50% accurate. And I'm going to come back to that later. But for now, I just want to look at that second question of which taxa are the most informative for constructing the random forest models. So I could obviously go into a huge amount of detail here. There were 140 random forest models, but I'm just going to give you a quick look at the overall ASV results because these gave on average the highest classification accuracy. Okay, so here we're looking at the average most informative taxa. So we have the taxonomic classifications shown on the right, and the average values for these taxa across all metadata categories shown on the left, with yellow at the bottom being the most informative taxa. And while there are other taxa that contribute to these models, I'm only showing the top most informative taxa here. When I talk about the informative taxa, the values that we use here are referred to as taxa importance, and that's the proportion that the accuracy of our models would decrease without that taxon. So then in the middle, we see how important each taxon is within each metadata category. So on the left, what we're seeing is how important the ASV is on average, and in the middle, it's how important the ASV is for each category independently. And we have all of the metadata categories along the bottom here, which are grouped by similarity. So just to give you a brief idea of the results here, we're seeing that usually for similar metadata categories, such as those based on material type, it's the same taxa that are most informative for that model each time. So it's the same taxa that we're seeing similar contributions from in each of these columns. And then, for example, if we look at something like salinity, so that's the furthest left category in this box here, then we see that some of the informative taxa are from the marine-only SAR-116 clade or the Alcanovorax genus. So hopefully I've demonstrated that overall environmental variables have a larger impact on the plastisphere than plastic type, and that we can identify taxa that contribute to these differences that biologically make sense. So now I'd like to move on to the other questions. So we're going to take a look at whether there are any taxa that are potentially specific to certain types of substrates, and then whether there are differences in the community composition between early and late incubation times. And for each of these, I've now split the samples by environment, and I'll be focusing on the marine environment. So let's first of all take a look at the differences between different substrate types. So now I'm only showing you the results from the marine environment. Here we've split the samples into 6 categories. We have aliphatic plastics in this greenish color, which is those such as polyethylene and polypropylene that consist only of long carbon chains. We have other plastics in purple, which is those plastics that have other functional groups such as polyethylene terephthalate and polystyrene. We have unknown plastics in blue, where the plastic type wasn't determined by the study. And then the controls, we have biofilm controls in orange, which included substrates like leaves, rocks, and glass. We have planktonic controls in yellow and methodological blanks, which are in grey, but there are so few that we can't easily see them on this relative abundance graph. So I'm grouping the plastics here because we otherwise have a huge number of really specific plastic types, but this means that we often don't end up with a huge number of samples within each of those categories. And if I just give you a quick look overall, here I'm showing a Venn diagram of only the taxa that are above 1% relative abundance on each substrate. And I don't want you to look at these numbers in any detail. I'm just trying to demonstrate that we have taxa that seem to be specific to each of our substrates, as well as not specific at all and present on all substrates, and then every combination of specificity to substrates in between that. And if we now construct random forest models for only marine samples and look at how well they're able to classify samples to different substrate types, they're now up to 83% accurate at the family, genera, and ASV phylogenetic levels, which if I just quickly show you the previous results again, where the same random forest model was constructed for all environments, where 46% is the maximum accuracy at the species level, then this is almost a 40% jump up in accuracy. And if we take a look at the taxa that are most informative for these random forest models, as well as those identified through ANCOM tests for differential abundance, then we start to see some key differences between plastics and other substrates. So here again, I'm not trying to show you in any kind of huge detail, but I'll point out what we're looking at right now. So if I just zoom in on this section so I can explain, we're looking at the abundance of taxa on our different substrates, with the categories being aliphatic, other and unknown plastics, as well as the control biofilms and planktonic communities. Here we have the taxonomic annotations along the bottom, which are colored based on whether the differential abundance was statistically significant or not. And we're also seeing the values for taxa importance within the models. And here we see that we have taxa like the hydrocarbonoclastic or hydrocarbon degrading ultramonadales that are significantly more abundant on other plastics, so things like PET or polystyrene than any other substrate type. We have the oceanospirales that are more abundant on aliphatic plastics than any other substrate type, so plastics like polyethylene and polypropylene. And then the halomonadaceae that are significantly more abundant on unknown plastics than any other substrate type. And if we look at these taxa that I've just mentioned, all of which are gamma proteobacteria, then we see that they've been known to be capable of the degradation of some similar substrates for quite a long time. Several members of the ultramonadales order, which was more abundant on the other plastics, can degrade crude oil, which is made up of a complex mixture of aromatic and non-aromatic hydrocarbons, not structurally dissimilar to the plastics that we're looking at, and for which the raw material in manufacturing is often crude oil. If we then look at the oceanospirales, which were more abundant on the aliphatic plastics, then many of the genera within this order are able to degrade alkanes, which is interesting because the aliphatic plastics, polyethylene and polypropylene, are essentially just really long chain alkanes. And then the family within the oceanospirales that was more abundant on the plastics of unknown type, the halomonadaceae, then these were again crude oil degraders rather than alkane degraders. This obviously doesn't tell us for sure that these taxa are degrading the plastics, but it's definitely suggestive that we might have some specific colonization of specific plastic types by taxa that are potentially able to degrade them. So I want to delve into the incubation time a little bit now. So what's immediately obvious here is that almost half of our marine samples were collected from the ocean after an unknown residence time, but then the remaining half, the mean incubation time is almost 100 days. So before I go into that time issue in too much detail, I want to give you a quick overview of what the degradation of a natural polymer looks like in the oceans. I'm just going to use the natural polymer chitin as an example here, because the colonization and degradation mechanisms of chitin have been comprehensively studied already, and it's something that I know really well because I've also studied it. And so chitin is the most abundant natural polymer in the oceans, and it makes up the shells of organisms like crabs and shrimps. And if we look at the microbial community succession across 4 days of incubation with chitin, we find distinct patterns in the abundances of different organisms. So here we're looking at the 16S and 18S rRNA gene communities, broadly speaking for the prokaryotes and eukaryotes respectively. And here we're looking at the relative abundance of these organisms. So for the 16S community, we see a huge peak in the abundance of these 2 organisms on the 2nd day, which is when enzymes to degrade chitin also peaked. And then we see a decline. And at the same time, in the 18S community, we see a huge increase in these 2 organisms, which are bactivorous marine flagellates or grazers. And these patterns of community succession and the colonization of chitin are really characteristic of microbial community ecology. We often see an initial colonization phase characterized by an abundance of organisms that are just good at attaching to particles. This is followed by a selection phase, which is characterized by high abundances of organisms that are actually able to degrade that polymer. And then a succession phase where these degraders are overtaken by cheaters, grazers, and viruses. So these cheaters are organisms that can't degrade the chitin themselves, but they can benefit from those that do by utilizing the subproducts of degradation. So the point I'm trying to make here is that usually when we look at the environmental degradation of a polymer, we need to be looking at our microbial communities at the right time if we're going to see communities that are capable of degrading that polymer, and not one of these communities that are just good at colonizing or the successional community. So are we able to identify taxa that are more abundant at early than late incubation times, which might be suggestive of their ability to degrade plastics? So here I'm again focusing on the aliphatic and the other plastics. We couldn't include the unknown plastics here because there weren't any samples where we knew the incubation time. And now if we first have a look at the aliphatic plastic samples, this is a tree showing significant changes in abundance between early and late incubation times, where early is below 1 week and late is above 1 week of incubation. So those branches that are yellow indicate taxa that are significantly more abundant at early incubation times, while red shows they were more abundant at late incubation times. And while this isn't strictly a phylogenetic tree, these branches are all ordered by phylogeny, just not by phylogenetic distance. So what's hopefully apparent here is that the taxa that are abundant at early or late incubation times are not just spread around the tree randomly. There's a clear phylogenetic pattern, which I also sulfocite in. And what we're seeing here is that the alpha proteobacteria are more abundant at early incubation times, and the gamma proteobacteria, Bacteroidea, and actinobacteria are more abundant at later incubation times. And then if we look at the other plastics, then we see a really similar pattern here, with the alpha proteobacteria again being more abundant at early incubation times, as well as the Verrucomicrobia in this case. And the gamma proteobacteria, Bacteroidea, and actinobacteria are again more abundant at later incubation times. So right now, this kind of seems almost contradictory to what we found on the natural marine polymer chitin. Here, the taxa that we think are potentially degrading the plastics, and the ones I pointed out previously are all gamma proteobacteria. They're all more abundant at later incubation times. This kind of leaves us with the question of what are early and late incubation times within the marine plastisphere. So before I move on to looking at those incubation times in more detail, I just want to summarize this part of the talk a little bit. We found that overall environmental variables have more impact on plastisphere composition, but there are taxa that seem to be specific to different plastic types. We've also identified some major knowledge gaps, such as how we have no information on the plastisphere in the southern hemisphere or in tropical or Arctic environments. And it's left us with some key questions that I want to explore for the rest of this talk. Can bacteria in the marine environment actually degrade plastics? Can they degrade plastic additives? Are the taxa identified here actually degrading the plastics? If so, did they use the same enzymes as terrestrial bacteria? And then finally, what are early and late incubation times within the plastisphere? So now I'm going to focus on whether plastics are being degraded in the marine environment. And as I mentioned before, perhaps the better question here is, can they be degraded? I focus this on polyethylene terephthalate, or PET, which is what plastic bottles are usually made of. And the reason why I've decided to focus on PET is because PET degradation has already been characterized in terrestrial bacteria. So we know that the key steps of PET degradation in terrestrial bacteria are the secretion of a PETase, a PET hydrolase, which is an esterase that has a high affinity for PET and breaks the PET down to MET or BET, which are both very structurally similar and are essentially the kind of monomeric subunits of PET. The MET is then degraded by a metase to terephthalic acid and ethylene glycol. And in the case of BET, this would just be terephthalic acid and 2 ethylene glycol molecules. And the degradation for both of these is relatively well characterized already. So here, by focusing on just one plastic, we were trying to address some of those previous questions on whether plastics for which the degradation is known by terrestrial bacteria could be degraded in the marine environment, which taxa might be degrading them, and how the issue of time plays into this. So the question we first looked at is, how does the plastisphere change across 6 weeks of incubation with PET as a sole carbon source? So what are those early and late incubation stages within the PET plastisphere? So I collected plastics from a beach in Wales, removed the biofilm, and I used this to inoculate a number of microcosms that contained PET powder, weathered PET powder. So these plastics were kept at 80 degrees for 6 months to hopefully initiate abiotic degradation. We have low crystallinity PET pieces. So crystallinity basically just refers to how tightly packed the chains of a polymer are, with lower crystallinity meaning that we have more of these amorphous regions with less tightly packed chains that are theoretically more accessible for microbial attack. And from these microcosms, we took a planktonic and a biofilm sample. And then we have the PET breakdown product BET and a no carbon control. So we incubated all of these microcosms for 6 weeks, taking samples on days 1, 3, 7, 14, 21, 30, and 42. And we sequenced the 16S rRNA gene communities, so the prokaryotic communities. So I first of all want to give you an overview of the microbial community on an NMDS plot. So here, points that are close together indicate that the communities are similar. And when they're further apart, the communities are less similar. So up here we have the inoculum, which is from plastics that I collected from the beach. Over here we have BET, that's the PET monomer, and it's semi-soluble, so it's therefore much easier to degrade. And then over here we have the low crystallinity PET, so that's the PET where the chains of the polymer are more easily accessible for microbial attack. And then we have everything else down here, which is only slightly separated from the no carbon control in yellow. So while we're kind of assuming that the grouping with the no carbon control means that these communities potentially just weren't growing very much, we wanted to know which taxa were driving the differences between these treatments. So to look at the taxa that were contributing the most to these differences, we performed a redundancy analysis that looked at each treatment compared with the control with no additional carbon added, and it tracked how much these communities changed relative to the control over time. So the control is effectively just the y=0 line here. So it's clear that, again, the biggest differences between the treatments and the no carbon control are with the low crystallinity, the amorphous PET biofilm community, which is shown in pink, and the BET community, which is shown in orange. And if we now look at the taxa that are contributing the most to these differences, then we see that it's different taxa that are driving the differences in each of these cases. And if we turn to look at the relative abundance of each of these taxa that are driving the differences, so here this heat map has each section being for a different treatment, and each of these across the 42 days or 6 weeks of incubation. So each column is from samples taken on a certain day. And then over here on the right, I've just got a blob representing the maximum abundance of that ASV and the taxonomic classification. And I'm just showing the BET and the low crystallinity or amorphous PET biofilm samples. But the preprint is available if you're interested in the other treatments. And what we can see here is that for BET, the taxa that are driving the differences are generally abundant at the early incubation stages. So that's these brighter yellow colors. Whereas for the low crystallinity PET, then they're tending to be more abundant at the later incubation times. And I'd also just like to highlight that some of the taxa that we're seeing here are within those hydrocarbonoclastic groups that we identified in the meta-analysis. So within the ultramonodales, which were more abundant on other plastics in the meta-analysis, which includes PET, we have 2 ultramonous ASVs that are most abundant on days 3 to 7. So these are the early incubation times that we classified before, and they reach a maximum relative abundance of 25% for ASV2, which is the bottom of these 2. And then within the oceanospirales, we have several taxa, but ASV8, Alcanovorax, reaches 15% relative abundance only on the last day of incubation, so after 6 weeks of incubation. So obviously this is interesting to see in terms of how the microbial community changes over time, and it suggests that we might be seeing some taxa that could be able to degrade the plastics. But this doesn't prove degradation by any stretch. So in order to have a better idea of whether members of the Plastisphere are able to degrade plastics, as well as how they might be doing this, we use PET as well as the PET monomer BET to carry out enrichments and then isolations of bacteria from our Plastisphere that could grow with these substrates as the only source of carbon. And from this, I managed to isolate 2 bacteria that show potential for PET degradation, Thioclava and Bacillus. So using also a previously characterized terrestrial PET degrader, Ideonella, as well as my 2 bacteria, I incubated these with a positive control, fructose, as well as terephthalic acid, BET, and PET. And you'll hopefully notice that these are all really structurally similar. So what we were hoping was that we could use these to really pinpoint the mechanisms being used for each step of the PET degradation. And we sequenced the genomes of these isolates, and we carried out both exo and cellular proteomics and metabolomics when these were grown with these 4 substrates. So through this combined proteogenomic and metabolomic approach, we were able to get this overall picture of PET degradation, and I'm going to take you through this step by step. So first of all, I'm going to talk you through some of the metabolomics results. And all of the substrates that we detect in metabolomics are shown as a fold change between negative controls with no bacterium and each bacterial incubation. And here I'm also showing the results from the final day, day 42 of incubation with our communities. So we're looking at this blue-green color scale. So light green indicates above 10-fold higher detection than in the control, and dark blue 10-fold lower detection than in the control. So if we first of all just zoom in a little on this part on the BET, here we've got from the bottom of the box the fold change for the incubations with terephthalic acid or TPA, BET, the PET monomer, and PET. And we're showing the results for Thioclava, Bacillus, Ideonella, and the community. So as we'd expect, we don't detect any BET in the terephthalic acid incubations. And in the BET incubations with Ideonella and Thioclava, it's perhaps slightly lower in detection than in the negative control. But in the incubations with PET, we detect a really large amount in the Thioclava incubations. And it's also significantly more detected in the Bacillus incubations, and that's shown with bold lettering, although it's not a lot more abundant. but it is also more detected with Ideonella, however not significantly so. So this is already a really great indication that PET might be being degraded by at least some of these isolates, because we're detecting this first expected breakdown product here. And now if I just extend the section we're zooming in on, we see that MET isn't detected with the PET incubations, but it is with the BET incubations, and it's actually most highly detected with the community, followed by Ideonella. Interestingly, in some of the incubations, we're also seeing these potential oxidized derivatives of MET, which to my knowledge don't have characterized degradation pathways. So perhaps we're not always following the exact same degradation pathways we expect from terrestrial bacteria. And then finally, if we look at terephthalic acid, then we see a lot building up in the Bacillus and Ideonella BET, but not PET incubations, but interestingly not in the Thioclava incubations. And when we incubate with terephthalic acid, we don't see any large changes in detection, which could be because it's toxic when added on its own, which is actually really similar to what we find for phthalic acid, which I'll get into a bit later on. So if we now turn our attention to the proteomics results, which I've shown for Thioclava in this pink to yellow, low to high color scheme, for most of the key steps in PET degradation, we're able to identify enzymes that are homologous to those used by Ideonella, our known PET degrader. However, in some cases, although Thioclava has proteins in its genome that are annotated with these functions, we don't detect them in the proteome. For example, the terephthalate dioxygenases that are usually used for terephthalic acid degradation exist in the genome, but we don't detect them in the proteome. However, we do find that other dioxygenases are much more abundant in the terephthalic acid treatment. So in these boxes, we're looking at the fold change in the PET treatment at the top, the BET in the middle, and the terephthalic acid at the bottom. So we think it's possible that these other dioxygenase enzymes might be capable of carrying out these steps instead. And likewise, protocatechouate, or 3,4-dihydroxybenzoate, is typically degraded by protocatechouate dioxygenases, but we only detect one of these subunits in the proteome, and it doesn't really seem to be any more abundant for the other subunit, while the catechol dioxygenases are up to 8,000 times more abundant. It's not really clear whether these are more promiscuous enzymes and they're capable of acting directly on the protocatechouate, whether they'd require the action of a decarboxylase first to convert this to catechol. And you might notice that with the proteomics results, I've only talked about Thioclava, and that's because while I showed you that we did detect them as metabolic intermediates in Bacillus, there were no enzymes in the genome or the proteome that were homologous to those used for PET degradation in our known terrestrial PET degrader, Ideonella. But what we do find is a really huge number of oxidases and monooxygenases that are all far more abundant when Bacillus is incubated with PET substrates than with our fructose control. This led us to hypothesize that perhaps rather than this pathway that both Ideonella and Thioclava use, maybe Bacillus just degrades PET through a lot of non-specific oxidation. So we incubated only the amorphous or the low crystallinity PETs with each of our isolates in our community, the same one that we used for the succession experiment, and we incubated them for 5 months. And then we cleaned up the plastics and performed FTIR spectroscopy on them to look for any changes to the structure of the plastics. And when we look at these, we're looking at the ratio of the peak at certain wave numbers of interest to a control peak that we know doesn't really vary. So for example, these bars are for the ratio of the peak at 1711 to the control peak at 1410. And this shows us when we have an increase in the number of carboxylic acid, carbon-oxygen double bonds on the plastic surface, which is indicative of hydrolysis of the PET chains. So the bars we're looking at here are the ratios I just mentioned. And we see that we have increases in the surface oxidation for PET that was incubated with both Thioclava and Bacillus in the blue and the red respectively, but the much larger increase in oxidation that we see for Bacillus is in accordance with the non-specific oxidation that we suggest might be taking place. And you might notice here that with the community in yellow, while some of these ratios are increased relative to the controls with no inoculum, this grey, none of the increases is statistically significant. So obviously it's possible that here we're not seeing any degradation by the communities at all, but we also think it's possible that because we're looking at a whole microbial community, any additional monomers or end groups that are generated might just be being used up by other community members really rapidly. And obviously this is just a hypothesis at this point, but degradation could therefore be more efficient by communities. And to try and investigate this hypothesis a little, we added some of the key enzymes for PET degradation that we found in our isolates to the PyCRUST-2 predicted metagenomes. And this revealed that we're predicted to have genes for enzymes for all of the key steps in PET degradation within our communities, as well as those other putative pathways that I mentioned. And the enzymes for all of these are often more abundant than in our controls with no carbon. And really intriguingly, if we look at only the phthalates, then all of the phthalates within the community can be attributed to just a few taxa. All of the taxa that are above 0.5% relative abundance and have phthalates belong to the Pseudomonadales, and each of these was predicted to have 2 phthalate copies in their genomes. So if we first look at this left-hand side, we're looking at the fold change of the phthalates with the no-carbon control communities across the 6 weeks of incubation. And we find them in the low crystallinity PET at the top, as well as in the PET powder and the weathered PET powder communities in the middle here. But if we look at the relative abundance of these phthalates, rather than the fold change, so this plot on the right, then they're only present in about 1% of our community members in the PET and weathered PET powder treatment. Only present in above 1% of our community members in the PET and weathered PET powder treatments. And their predicted abundance increases over time, which suggests to us that in 6 weeks of incubation, we haven't actually yet reached the peak of PET degradation. So to summarize this section a bit, I just want to try and tie this back to our original questions. Can bacteria in the marine environment actually degrade plastics? I'd say the answer here is yes, they can, but the results I've shown you here were from experiments performed in the lab, and they're also only for one type of plastic. We don't know how much plastic degradation will be affected by other environmental conditions, and so we definitely need some more work there. Secondly, is it the identified taxa that are degrading plastics? Well, the 2 bacteria that we isolated weren't from the hydrocarbonoclastic gamma proteobacteria groups that we were originally looking at, but we did see those hydrocarbonoclastic groups again within the community incubations. So it's clear that we still need to do some more work looking at these taxa specifically to find out if they can degrade plastics, and particularly whether the plastics can be degraded in the context of a community incubation. Do they use the same enzymes as terrestrial bacteria? Well, yes and no for Thioclava and Bacillus respectively. There obviously needs to be some more work looking at the mechanisms used by our Bacillus species before we can answer this for sure. And until we've identified exactly which enzymes are used for degradation, it's difficult to say whether they just share very little homology with the characterized enzymes or whether they might represent a totally new pathway. Either would obviously present the chance to search environmental metagenomes for the presence of these enzymes in other species, though, which could give us further insight into environmental plastic degradation. And then finally, what are early and late incubation times within the plastisphere? Well, again, we really need more information to be able to answer this, but it seems like this probably varies for each plastic, because even within just polyethylene terephthalate, we see differences between the different degrees of crystallinity and also with the monomer as opposed to the polymer. So now I'm going to move on to that final question of whether marine bacteria can degrade the additives of plastics. So the objective here was to see not only the degradation of the polymer itself, but also the degradation of additives of plastics. So plasticizers are chemicals that are added to plastics during manufacturing in order to improve their physical properties. And in many cases, they're what makes the plastic plastic, because they give the plastics properties like flexibility and durability. And for example, in PVC, up to 50% of the weight of the plastic can be made up of these chemicals. Many of them are known to be endocrine disrupting, carcinogenic, mutagenic, and even deadly. And because they're not chemically bound to the plastics, it is now well documented that they can leach out from the plastics and become available not only to the environment, but also to microbes that might be able to degrade them. So our objective here was to identify who is degrading and how are they being degraded in the marine environment. So I chose to work with 2 toxic plasticizers, which together make up about 65% of the plasticizer market, DEHP or bis-2-ethyl hexylphthalate and DBP, dibutyl phthalate. These are both phthalic acid esters, and their degradation in terrestrial environments has been known since about the 1950s. And the mechanisms for degradation seem to be well characterized, although the complete degradation of the chemicals is still relatively slow. I then also chose one plasticizer that's widely stated to be a good alternative to these because it's biodegradable and also less toxic. That's ATBC, or acetyl tributyl citrate, which is the citrate plasticizer. However, despite the fact that several studies that use ATBC state that it's a biodegradable plasticizer, I wasn't actually able to find any study that showed its biodegradability previously. So we wanted to see if the mechanism for degradation would be the same for these two phthalic acid esters, and then if the ATBC really was biodegradable. So I originally isolated 42 bacteria that were able to grow with these plasticizers, as well as 3 others. And here I looked at whether there was clear growth, weak growth, evidence that the plasticizers had been emulsified, even if we couldn't measure growth, or whether there was no indication of growth at all. And interestingly, about 3/4 of the isolates showed some indication of being able to grow with both DEHP and DBP. But for ATBC, the supposedly more environmentally friendly and biodegradable plasticizer, only about half of the bacteria could grow with it, and only 1 bacterium actually showed really strong, obvious growth. So we narrowed the 42 isolates down to 10 that showed good growth with more than 1 plasticizer, and then chose 2 that could grow with all of them, Halomonas and Mycobacterium. And if we look at the growth curves for each of these bacteria on the plasticizers, as well as phthalic acid, which is just the red structure that I've highlighted now on both the DBP and DEHP. So we have Mycobacterium in blue and Halomonas in this peachy color. We can see that both bacteria show reasonable growth with the labile substrate control on the left, and each of our 3 plasticizers on the right, but not with the phthalate or phthalic acid, as it's also known as. So we had a few specific questions that we wanted to investigate here. Firstly, as we couldn't find any previous study looking at ATBC degradation, how is it degraded? I mean, is it even degraded? Secondly, does the degradation of our 2 phthalic acid ester plasticizers follow the same pathways as have been previously identified in terrestrial bacteria? Are the side chains from different plasticizers removed using the same mechanisms? So hopefully you can see here that both dibutyl phthalate and acetyl-tributyl citrate have these butyl side chains. So we wanted to know if the same enzymes will be used to remove the butyl side chains from 2 different molecules, and then whether the mechanisms for side chain removal will be the same between the longer branched chains of DEHP as the DBP. And then finally, why do we not see any growth with phthalate? So to answer these questions, we again carried out a thorough proteogenomic analysis through sequencing of their genomes and carrying out proteomics and then also metabolomics. So I carried out this out with these 3 plasticizers, as well as also phthalic acid. And this proteogenomic approach gives us this overall pathway of marine plasticizer degradation. I know this is a really busy figure, but I'll talk you through all of it now. So first of all, while you probably don't really need to know this in detail, I just want to explain exactly what you're seeing here. So all of the initial substrates are in grey boxes. The coloured substrate names means that we detect this substrate with metabolomics. So here this MBP or monobutyl phthalate is detected in the metabolomes of Mycobacterium, the blue background, and Halomonas, the peachy background. And we always have the structures of these compounds close to their name. In these diagrams, I haven't indicated which incubations we detect these intermediates in, only that they're detected, but we only ever grew each bacterium with 1 plasticizer at a time and not a mixture. Then we have our proteomics results in these boxes next to the arrows. So each one is for 1 protein in Mycobacterium on the left and Halomonas on the right, along with the genomic coding domain. And we have the results for the fold change between the abundance of that protein in the control and in each of our treatments, with dibutyl phthalate at the top, DEHP in the middle, and ATBC at the bottom. The dark red here means that this protein was at least 10-fold more abundant than with our labile control, and dark blue would mean that it was at least 10-fold less abundant. So if I first of all just show you the metabolomics results, you can see that already we're filling in a lot of our pathway, but a lot of these intermediates are only detected in 1 rather than both bacteria. And if I now focus on dibutyl phthalate, this phthalate degradation pathway shown directly underneath the DBP is what's found in terrestrial bacteria. And we find all of the metabolic intermediates for this pathway in Mycobacterium, but while we find the first steps of it in Halomonas, there's not all of it. So first of all, the butanol side chains are removed, producing first monobutyl phthalate and then phthalate. And it's esterases that are likely to be used for this. And these were significantly more abundant in both the Mycobacterium and the Halomonas proteomes. But you might notice that in Mycobacterium, the esterase we've identified is only abundant in the DBP treatment, whereas in Halomonas, it's abundant in all plasticizer treatments, but particularly so in the dibutyl phthalate and the ATBC treatments. And if we now look at phthalate degradation, we see that these proteins are particularly abundant in Mycobacterium, again, specifically in the dibutyl phthalate incubations at the top of the boxes, but they're not for Halomonas. And if we look in the genomes of these isolates, we see that in Mycobacterium, all of our phthalate degradation genes are in a cluster in the genome, along with 2 transcriptional regulators. And we detect all of these proteins in the proteome, so shown in red, aside from one of the transcriptional regulators, which is shown in grey because it wasn't detected in the proteome. And if we map these proteins onto the dibutyl phthalate or phthalate degradation pathway, then we can see that we've got proteins for the whole pathway that are detected in the proteome. And this was the bacterium where we also detected all of the intermediates with metabolomics. But if I now jump over to Halomonas, then we have this huge region of the genome running from coding domain 593 to 691 that is dedicated to monoaromatic compound degradation, with a lot of it detected in the proteome, so shown in red. But if we try to map this onto the degradation pathway, then it gets a bit more complicated. So if I just point out phthalate here, then we don't find all of the genes that are needed for that standard phthalate degradation pathway, and they aren't elsewhere in the genome either. But what we're seeing is that we potentially have a lot of other pathways with very similar compounds that might be being used instead. We just aren't quite sure whether these enzymes will be able to act directly on the phthalate or need something else to convert them into other substrates first. So while we would obviously need to do further tests to confirm this definitively, the proteomics along with the metabolomics not detecting any of the standard phthalate degradation intermediates suggest that Halomonas is using a different pathway for phthalate degradation, and perhaps even more than one pathway for it. I now just want to quickly turn your focus to phthalate here. So I've just been talking about the degradation of phthalate, but that was phthalate that came from dibutyl phthalate. But what happened when we just added phthalate to the bacterial incubations? As I showed you before, we saw practically no growth with phthalate. And we didn't really find any of the proteins needed for phthalate degradation in the proteomes either when we were growing with just phthalate. But what we do find are a number of proteins related to stress responses and cellular detoxification mechanisms, as well as this huge buildup of phthalic anhydride. So we did a quick experiment to test out the toxicity of phthalate. So we grew both bacteria and marine broth, so an additional carbon source, and we added different concentrations of phthalic acid from 0% shown in black to 1.6% shown in blue. And we have Mycobacterium on the left here and Halomonas on the right. So you can see that both bacteria grew with up to 0.02% phthalic acid with marine broth. So we thought that maybe we needed something different to induce the phthalate degradation operons. But intriguingly, we find that when we add dibutyl phthalate or DBP, where we saw really strong induction of the phthalate degradation, particularly for Mycobacterium, then phthalate toxicity actually increases as we now don't have growth with even the lowest phthalate concentration. So we assume that we have small amounts of phthalic acid as produced when degrading dibutyl phthalate. And when we have this, it doesn't inhibit growth. But as soon as there's more, then these bacteria aren't able to cope with the phthalic acid. And they seem to be converting as much of it as possible to the phthalic anhydride, which is less soluble than phthalic acid and would therefore presumably reduce the toxicity to the bacteria. And then to just finish off this dibutyl phthalate story, the butanol that's removed from the DBP molecule to produce phthalate is degraded through the short-chain fatty acid beta-oxidation pathway, and we detect a lot of these proteins. We don't find any of the metabolites, but they likely wouldn't be detected by the metabolomics conditions we used anyway. So now I'll just take you over here to DEHP. So the studies on terrestrial bacteria that I mentioned previously had suggested that degradation would be through an initial loss of the side chains producing phthalate. But here we didn't detect any phthalate at all in this treatment, or any of the other phthalate degradation intermediates. And as I mentioned before, we also didn't find that these proteins were any more abundant with DEHP than our control or any other treatment. So we don't know whether the side chains are lost from DEHP, and if they are, then they don't seem to be producing phthalic acid. But we detect a lot of proteins from the long-chain fatty acid beta-oxidation pathway. And we think it's possible that these enzymes are actually acting directly on the DEHP side chains, progressively shortening them and funneling them into the TCA cycle. And we see that particularly for some of these proteins in Mycobacterium, they're far more abundant in the DEHP treatment than the control or any other treatment. So now I'm finally going to take you over to ATBC degradation. So this is the plasticizer that's been hypothesized to be less toxic and more biodegradable than the other plasticizers, but we hadn't been able to find any previous study that showed that it was all the mechanisms that were used for its degradation. So we are detecting some of these metabolic intermediates in the Halomonas metabolome and some in the Mycobacterium metabolome. But what these are suggesting is a sequential removal of butanol and acetate to produce citrate. And again, we probably wouldn't be able to detect the isocitrate succinate or glyoxylate with the metabolomics conditions that we used. And these first steps are, as in dibutyl phthalate degradation, carried out by esterases and carboxyl esterases. But it seems that we probably have some slightly different induction of some of these enzymes than we were seeing with the dibutyl phthalate. And then we see that we have enzymes for the conversion of citrate to isocitrate, and that the enzyme responsible for the conversion of isocitrate to succinate and glyoxylate, an isocitrate lyase, is actually making up 25% of the relative abundance of the exoproteome, and is 34-fold more abundant than in the control for Mycobacterium. So we're fairly confident that although we don't detect these intermediates, this is how the degradation of ATBC is happening. And then while we don't find that these fatty acid beta-oxidation proteins are hugely more abundant with ATBC than the labile control, as they were for DBP, they are detected. But, and I'm just kind of speculating here, butanol potentially isn't as essential for growth when you're also producing the succinate and glyoxylate that can go directly into the TCA cycle. And this gives us this overall picture of plasticizer degradation by marine bacterial isolates that I hope I've managed to explain well enough to you. So can marine bacteria degrade plastic additives? Yes, we found several isolates that can degrade these additives. Our study was the first to show ATBC degradation and also to show the mechanisms used by different Plastisphere members for additive degradation. We show that an array of esterases are involved in the first steps of both dibutyl phthalate and acetyl tributyl citrate degradation. The induction of catabolic pathways for plastic additive degradation is not always straightforward. Different mechanisms are used for the degradation of side chains from different phthalic acid esters, and a number of strategies are used by the microbes to deal with these toxic compounds. So I hope that what I've shown you here is an overview of what we already knew about the plastisphere. There are sometimes bacteria that are specific to different plastic types, but the environmental conditions overall have a larger impact on microbial community composition. When we look at the plastisphere of just one plastic in more detail, we see that the different stages in community succession appear to occur at different times, depending on the plastic in question. But we can isolate bacteria that are capable of plastic degradation, with one of these bacteria using the same pathways previously characterized in terrestrial plastic degradation, while the other potentially uses an as yet uncharacterized non-specific oxidation. And then finally, members of the plastisphere are capable of the degradation and detoxification of plastic additives, but the specific mechanisms that they use may vary a lot between different bacteria and different plasticizers. So are plastics degraded? Well, I hope I've shown you that they definitely can be, but we still have a lot more work to do before we can confirm whether this is really happening in the environment or not. So I'd just like to thank everyone that I was working with while I was in the UK, my PhD supervisors, Joseph and Matt, as well as everyone else in their groups, and also Raphael in Spain, and my funders at MIBTP. I'd then also like to thank everyone over here in Canada that I've been working with for the last year, Tony and Morgan, as well as the rest of Morgan's group, and the ACU for funding my meta-analysis. And if anyone has any questions, then I'd be happy to take them now, or do feel free to get in touch with me later on also. Thanks.
Yeah, Robyn, this was really amazing. It was really like a mind-blowing talk. And thanks for all the details on the microbial metabolisms.
Thanks for having me.
So yeah, I guess our Californian colleagues are not up yet, but it's good that you put your contact here so they can ask their questions later. I had a question about the evolution of plastic degradation. So I was wondering, are there places in the ocean where microbes have come into contact with crude oil since like thousands of years, and where we could learn a little bit more on what kind of pathways they evolved?
Definitely. So, I mean, a lot of the bacteria that I pointed out, I mean, often they're bacteria that are kind of ubiquitous in the ocean, and we see huge blooms of them when there's an oil spill, for example. And this is where a lot of the focus on any potential plastic degradation has kind of gone on to. It's just that we're still kind of lacking in a lot of studies that actually really fully characterize this degradation and really pinpoint the mechanisms used, and also kind of definitively prove that the plastic really is being degraded. It's not just fragmenting into smaller pieces or something.
Yeah, but when you say oil spills, you mean like human-caused oil spills?
Well, I mean, I guess oil spills, or it could just be where there's oil plumes in the oceans anyway. Mm-hmm. Either way, I mean, it's going to be, I guess, the same bacteria that are probably doing that degradation.
Yeah, I'm just asking because the human-caused oil spills, they're not so old.
No.
And then I was also wondering about the microbial ecology when you talked about the timing of colonization. Mm-hmm. And I was wondering when you showed that slide with the cheaters that come up during like chitin degradation, how do you define a cheater? What do they actually do?
So I think some people probably define this differently, but personally I would define it as a bacteria that can't degrade, so in the example of chitin, that can't degrade the chitin itself, but might be able to use the monomeric subunits of the chitin for growth. But then some people also look at the cheaters as kind of organisms that maybe actually can't even use any of the monomeric units of the chitin either, and they're just able to use the kind of organic matter generated by a community.
Okay, but then in a way they're not really harming like the first colonizers, they're just taking their products.
Well, it depends if it's products that the degraders can't use or products that the degraders wanted to be able to use for growth, because especially when you're looking at the degradation of a polymer, they obviously can't take the whole polymer inside their cell to grow with. So they're initially breaking it up outside of the cell, and then perhaps if those products are being used by something else before the original degrader can use them, then the original degrader can't grow as much.
Yeah, that makes sense. And then another thing that I think you mentioned is that you didn't find much data about the plastisphere in the tropics and in the Arctic. And I was a little bit surprised why there's no data there, because I thought there were like a lot of microbial ecology groups like Chile and Argentinian, and also like in like the Smithsonian in Panama. And I was just wondering, so is this like something that you observed with your meta-analysis, that these areas are just not well sampled?
Yeah, so I mean, I guess maybe I should be a little bit more specific that it's that there aren't studies that use next generation sequencing to characterize the plastisphere in these environments. But given the kind of explosion in studies that use next generation sequencing in the last few years, and I mean, I think there perhaps have been a couple this year. So since I had kind of worked on the meta-analysis that are now looking at this. But definitely all of the work that's kind of the first 10 years of the plastisphere, I suppose, using next generation sequencing methods, it hasn't included the tropics or the Arctic.
Oh, I see. And also I was wondering about from your, like the data from your meta-analysis, whether you could use that data and look into dispersal of microbial groups. So for example, whether you could ask whether plastics facilitate the dispersal of like some given ASVs, and whether you could test that with that data.
Yeah, so I mean, that was kind of something I'd wanted to get into, but some of the problem is that often studies have looked at plastics and they've looked at planktonic communities, but if they don't have kind of another biofilm control, then it's really difficult to say whether it's the plastics that are causing something to be spread, or whether it's just, you know, biofilm forming organisms that are there. I mean, plastics obviously persist for longer and potentially are transported much further than you might expect something natural to be. But I mean, that hasn't really been looked at. And I mean, I guess it's a really difficult experiment to really properly do. I think there's now this year been a couple of studies that have looked at plastics as they come down a river and out to sea, for example, and looked at how this affects the change in the community. But yeah, hopefully there'll be some more studies on that. It'll be really interesting to see.
Yeah. And then I was wondering, with your incubation experiments in Wales, when you looked at plastic degradation, I was just wondering, how fast does this happen? Like, can you give us some kind of ballpark numbers about, for example, if you have a PET bottle, how long would it take to like degrade a whole bottle?
I mean, I didn't look at calculating that. I think I've seen something else maybe this year. It came out, and I think they guessed at 50 years or something for a microbial degradation of plastics. I mean, it's still really slow, especially because generally, I mean, in the first studies that found terrestrial bacteria that degrade plastics, as well as in what I looked at, we're using lower crystallinity plastics to try and speed up so that we can actually look at this degradation. But the plastics that are in the environment are much higher crystallinity and then therefore a lot more difficult to degrade. So, I mean, I would love for, I guess, bacteria to be the solution to everything, but I don't think that's the case at all.
Yeah.
It's still going to be pretty slow.
Yeah. So yeah, I think these were all the questions. And I would like to thank you again.
Thanks for having me.
Yeah, it was fun having you. And yeah, please, if anybody's seeing this later, just reach out to Robyn.